Memory is an important part of being human. I participated in a research program that looked at how memory can potentially be affected by certain proteins at the molecular level in the human brain. The part of the brain that's responsible for memory is called the hippocampus. It has many regions in it. The one of interest is called CA1. It contains thousands of neurons that connect with each other. They do this with the help of special proteins called guidance cue proteins. They can either encourage or inhibit connections between neurons. Now there's a specific guidance cue protein called SEMA3A, and it's very obscure because there's very little research on it so far. What we already know is that it is present in the human brain during embryonic development, but what we do not know is if it's still present in a fully formed adult brain. So in this experiment, we try to discover its presence in an adult mouse brain. So how do we do this? Well, to start, I didn't actually look at SEMA3A itself, but instead I looked at something called its receptor. Every protein has its own specific receptor that it binds to in order to start its chemical reaction. So for SEMA3A, part of its receptor is made up of this molecule called neuropilin. So I focused on visualizing neuropilin in the hippocampus, and I did this with a technique called immunohistochemistry. So I took very, very thin slices of adult mice brains and put them in different solutions to allow fluorescent tags, or neon colors, to basically bind to neuropilin, and this way I can put these brain slices under a microscope and get these pretty images. This image is zoomed into a couple neurons in the hippocampus. These green blobs actually indicate the neurons themselves, and the green lines coming out of them are the connections that are being formed. So what we see in this image is a close-up shot of the CA1 region of the hippocampus. Neuropilin is stained in the color yellow, and the pink lines indicate excitatory connections between neurons. So what we see here is that neuropilin is in close proximity with these excitatory connections. This research and these images have shown that SEMA3A is in fact still present in a fully formed adult brain.